This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, host of the show. Cell and developmental biology is a field that's integral to finding new therapies for a wide variety of diseases. Here at Feinberg, Dr. Lusia Iruela-Arispe, a vascular biologist, leads the Department of Cell and Developmental Biology as chair. She joins me today to talk about her research and the future of cell-based treatments for diseases. Welcome to the show.
Hi, Erin. Thank you for having me.
Well, tell me about cell biology and the ways that the field has grown in recent years to impact clinical practice and therapies for sick patients.
Well, cell biology is an amazing discipline. It's one that really encompasses or is part of many others like biochemistry, genetics, epigenetics. Everything is within cell biology. And so that's why I'm so passionate about this particular area. And it has really grown tremendously, I think, in particular, how it impacts cancer biology. If you think about all the chemotherapeutic drugs that we have today, they are mostly affecting cytoskeleton elements and the process of cell division in cancer cells. So it has been through understanding cell biology that we have made so many advancements, particularly in cancer, but not only exclusively in that area.
Tell me about some of the human diseases that you're trying to improve through work in your lab.
Our laboratory is a vascular biology laboratory. I'm really very passionate about blood vessels. They are tubes that permeate our body, but in reality, they impact both healing as well as they can accelerate or really make certain steps in disease progression worse. So by manipulating blood vessels, we can impact either the healing process or accelerate or even eliminate certain diseases. So it is really the route by which inflammatory cells traffic. So everything that relates to inflammation has to do with enabling inflammatory cells to leave the bloodstream and access the organs in general and affect disease. So by regulating that process, we can definitely control inflammation, but we can also control the trafficking of, let's say, cancer cells to prevent metastasis.
How did you first become interested in blood vessels and studying the vascular system at this molecular level?
I remember this vividly. I was a graduate student and I was hearing about this cell type, endothelial cells, which are the cells that are present in all our blood vessels. They line the inner side of all blood vessels. And these cells normally will just be responsible for transporting molecules and nutrients from the blood into our tissues and vice versa. So they act as a transporter, if you will, and they will allow cells to pass through and one to the other. But the neat thing is that they can also completely change and undergo a rapid process of proliferation to form new blood vessels. So they have this plasticity that is very unique to, I think, endothelial cells, in which they can be fully differentiated or become as active as an embryonic cell just with a change of a dime, just by having a growth factor activated them. I think that you can actually utilize them obviously to form new vasculature and permeate new structures. So we are now in the area of regeneration biology. And by creating new organs or organoids, we can replenish damaged tissue. We can definitely impact disease in a very positive way. But these organoids require vasculature. And so the other thing that you can do with the cells is you can enable them to integrate with other cell types to permeate structures and provide nutrients and oxygen. So you can essentially 3D print them, you can manipulate them, and you can integrate them with other cell types to generate new tissues.
That brings me to this idea of bioprinting. This is an emerging area of study which uses biological materials to create these tissue-like structures that imitate natural tissue. How involved are you with this research right now?
We definitely do plenty of bioprinting. In fact, we have quite a few in the laboratory. So what we're doing is trying to integrate again organoids, brain organoids, intestinal organoids, and make them permeatable with blood vessels so that they can grow, expand, and better imitate, mimic normal organs. So that has been a fun project.
You are able to work with so many different departments here at Feinberg, and both on the clinical side and basic science. Tell me about some of these partnerships and some of the diseases that you're able to work on.
There are several collaborations that we have going on. I mean, one with the Department of Pharmacology, Al George's department, being able to manipulate through pharmacological means the function of any cell type is one of the bases of how to control disease and how to control health. So one of the areas that Al works a lot with is channels, ion channels, which control essentially flow of ions as well as a lot of properties of the cells in general. We have been collaborating with him very actively to understand how manipulation of those channels can actually improve vascular health. That's one example. We also collaborate with the Department of Neurology quite heavily. That has been also very interesting and fruitful, and it takes us into the area of vascular dementia and how to improve the vascularization of the brain and what goes wrong when in cases of Alzheimer's and other situations that compromises the vasculature as well.
Tell me about this idea of cell-based therapies. What is happening right now, and how close are we to having more cell-based therapies available to patients?
The area that initiated this in a big way is hematology. So transplantation of bone marrow to replace bone marrow of patients that have mutations that lead to leukemia has been transformative. And this is something that for blood is a lot easier because we have a way of delivering. And these cells, once we understood what was needed, they are professional. They know where to go and how to expand. And they're able to colonize in a way that substitutes disease for healthy bone marrow. So I think that we learn a lot from that process. And this idea now of how to use new cells to substitute for all cells has taken over. Now, because most cells are not circulating and they don't have the same properties of hematopoietic cells, it's become a little challenging. And that's why the development of organoids has been so interesting to biologists and developmental biologists and cell biologists. And I think that that is going to be probably the future of regenerative medicine.
Tell me about your recent article in Nature Cardiovascular Research. Your team discovered a new cell type that was not previously understood. Tell me about this exciting finding.
So this is a new type of macrophage, which is a professional, should we say, inflammatory, but also more like a cleaner cell type that lives in the endothelium. So I mentioned to you, right, that the endothelium is this layer that is responsible for covering the inner surface of all the blood vessels. Normally, that area only is comprised of endothelial cells, but we found through this study that was recently published that there's a new type of macrophage that is present and intertwines with these endothelial cells in areas of oscillatory flow. So let me take back this a little bit. So because some vessels are twisted and turn, when blood goes through them, it creates turbulence and creates disturbed flow. So think again about pipes, and when pipes divide, what happens to the flow? It turns out in those areas of turbulent flow, there is accumulation of thrombin, which results in clots. And so the function of this macrophage that we discover is to eliminate those clots. And that's very important because when we experimentally deleted them, when we remove them from these areas of turbulent flow, the vessels get clogged. So they get full of fibrin and they have intravascular thrombosis, which is obviously very problematic. That was a very exciting manuscript and exciting paper because it told us many things about the endothelium, and also it showed that we need to have these professional cells in areas of turbulent flow to avoid clots from being formed.
Why is this finding so timely?
Yeah, I mean, I think the reason why that was timely and so interesting is because of COVID. So there is this concept and this idea that one of the consequences of COVID-19 is to have problems with blood vessels. And that is why the long COVID is affecting so many systems. So When these macrophages are affected, then intravascular thrombosis is a lot more frequent. And if you have intravascular thrombosis in small vessels in particular, you can have clogs and ischemia. So this is an ongoing investigation, obviously, and something that we are expanding significantly. But it can have very significant consequences to understanding disease and understanding how disturbance of one particular cell type can trigger consequences that are system-wide.
So what has the reaction been like from the scientific community to this finding?
It was quite interesting. Obviously, the way that we perceive those reactions right now is through Twitter. It's interesting to see, right? That's essentially the pulse of what the impact of a discovery like this has been. And it was very active. We had a lot of very positive feedback. But it's a process. And one discoveries are only important if we communicate them to others and others walk them further. So this is going to be a process that I'm looking forward watching and continuing to participate, obviously.
You also collaborated on an article in Nature with a former colleague of yours from UCLA that expands our understanding of how blood cells and endothelial cells crosstalk. Share those results with me.
So as you mentioned, I'm a co-author. It required the effort of multiple laboratories, and the senior author is my very dear friend, Hanna Mikkola. She is at Cell and Developmental Biology at UCLA. And what Hanna has been very interested in knowing is how to improve the generation of hematopoietic stem cells in vitro. But for that, we needed to go in vivo. So as we learn, and in fact, many years ago, we published this, hematopoietic cells come from endothelial cells. And so they bud off endothelial cells during development. So my contributions were to provide this understanding of endothelial cell biology and understand how they actually bud off from endothelial cells and learn the molecular intricacies of this crosstalk between endothelial cells and hematopoietic cells that are budding off. And what Hanna did is took this forward, and essentially she identified the key molecular drivers that can inform how we can better do this in vitro so that we can use in vitro systems to form hematopoietic stem cells and use them for transplantation.
This is sort of like step one. So what comes next?
Yeah, so with this information that she just obtained and communicated to the community, she is now developing new systems in vitro to do just that. So it's incredibly challenging because, again, the beginning requires this crosstalk between two very different cell types and cell types that are at a different stage, right, than what we normally use. So she needs to take them back to that time, that embryonic time, take them back, meaning molecularly by disturbing them in vitro, and recapitulate or mimic that genetic time, that differentiation time, so that we can actually provide benefits to millions of people that are affected by blood diseases or blood cancers, or perhaps even regeneration of blood.
You actually took on your role as chair of the Department of Cell and Developmental Biology not too much before the pandemic started, but tell me about your role as chair and some of your goals for the department and what you've been able to accomplish so far.
That's a big passion of mine. I mean, trying to shape a department like Cell and Developmental Biology at Northwestern is a privilege, and I'm very, very excited about what the future waits. So when I took on the department, you're absolutely right, it was like months before the pandemic. And so obviously a lot of our plans were curtailed by what followed. But nonetheless, the vision was articulated very clearly to all the members of the department, and it was shaped further by them as well. So I had the opportunity to discuss with them how I view the direction of the department in the next 10 years. And the pandemic allowed us to have a more extensive conversation as to what are the directions and the path that we should go. And essentially, we all agree that what we want is to build a department that is going to be the top department in the nation in the next 5 to 10 years. So the question is, where is the discipline going to be in 5 to 10 years? What are the challenges that we are going to encounter? And where are we going as scientists in this area? So answering that question is kind of, you need to have your little glass ball and understand where are the key directions. And I think that is very, very obvious that computer modeling, quantification, math, and integration with physics as well as biology is going to partake a very important role in the advancement and development of this discipline. So we want to hire people that can walk both walks. They feel comfortable with the idea of using modeling to develop hypotheses and then test those hypotheses in what we call a wet lab in the laboratory with Petri dishes and pipette mints. So we want real biologists, but those that are also able to expand, understand, and if not, embrace. Like, for example, a dinosaur like myself. I'm not a computer scientist, but I definitely appreciate, respect, and I would love to collaborate with those who have those abilities to further our disciplines. So we are trying to identify raw talent that is able to embrace this vision and that hopefully has those elements already built in and incorporate them into the department. So I think that despite the pandemic, we have been able to actually move things forward in the same path as we intended originally.
Thank you so much, Dr. Luisa Iruela-Arispe, for coming on the show, telling us about some of your recent discoveries and how you are leading your department. We really appreciate it.
Thank you, Erin. I really appreciate it too. Thank you.
Thanks for listening, and be sure to subscribe to this show on Apple Podcasts or wherever you listen to podcasts, and rate and review us. Also, for medical professionals, this episode of Breakthroughs is available for CME credit. Go to our website, feinberg.northwestern.edu, and search CME.